Hello, and welcome to our program this evening. I'm Stephanie Gaspar, president of Kissimmeevalleyaudubonsociety, and appreciate everyone watching our program. Larry Rosen, one of our current board members, will also be joining the panel with us this evening. We are happy to have him. Last month, Paul Eisenbrown was our guest speaker. He shared a lot of great pictures of birds and animals from around Central Florida. I'm glad he mentioned many of the different birding hotspots that folks can visit. I recommend you visit some of the locations if you can. I also have good news. Our board of directors have decided to add a program for next month. Rainier Munguia will be our May guest speaker, and the topic will be all about woodpeckers. Additionally, we are seeking members to join our board. There are various open positions, so please get in touch if you are interested. Stay up to date with our programs by checking our website, following us on Facebook, and be sure to subscribe to our YouTube channel. Before we begin, I would like to mention a few housekeeping items. Log in to YouTube and type any questions you have in the chat. We will wait till the end to answer all questions. Tonight's topic is very interesting. Backyard chicken coops are increasingly popular, but can backyard chickens cause disease in wild birds? This evening, I am pleased to present our guest speaker, Dr. Andrea Ayala. She'll present the results of her research on viral transmission of Newcastle disease between backyard chickens and wild birds in the Athens, Georgia area. Andrea is an NSF Postdoctoral Research Fellow at Yale University. She earned her PhD from the University of Georgia's College of Veterinary Medicine and a master's in ecology from the Odum School of Ecology. Thank you very much for joining us today, Andrea. We are excited to have you, and I will now pass it over to you.
Thank you for having me, Stephanie. I'm very honored to be here. And I'd like to thank everyone for coming. I'd also like to thank my co-authors for their contributions through this journey. They're listed at the bottom of this slide, and this dissertation just would not have been possible without them. So today's topic is, do backyard chickens pose a pathogen risk to wild birds? And I have a titmouse here because we're going to be revisiting this species quite a bit later on in the presentation. And so here's the agenda. Because we'll be covering a lot of information as I go along, I won't get into excruciating detail here of what we'll be covering. So this topic and this experiment was inspired by my master's, in which we incidentally found evidence of a Newcastle disease virus vaccine in rock pigeons of Atlanta, Georgia in 2012. And there I am banding a rock pigeon in downtown Atlanta. Originally, I set out to examine how co-infection with gastrointestinal parasites influenced the viral shedding of pigeon paramyxovirus, a strain of Newcastle disease virus. For those of you that don't know, Newcastle disease virus is an economically significant pathogen of poultry, with 78 countries reporting outbreaks in 2018. It's reportable to the OIE, also known as the World Organization for Animal Health, and the only other virus that is reportable to the OIE is avian influenza, which shows how significant this pathogen is. So finding these vaccine viruses in rock pigeons of Atlanta, Georgia, caused me to ask the question, how does spillover between domestic and wild birds occur? And specifically, how do these groups come together to enable pathogen transmission? So I was inspired to examine the backyard chicken wild bird interface, and I learned some very important points. Pathogen transmission dynamics at the backyard chicken wild bird interface are bidirectional, and they're subject to ecological and evolutionary pressures. When backyard chickens share habitat with wild birds, natural contact barriers are often lost. That loss of contact barriers may facilitate the transmission of opportunistic generalist pathogens. Importantly, backyard chickens are increasing in popularity, and between 2 and 7% of households plan to own backyard chickens in the next 5 years. So given that it's impossible to examine all potential pathogen transmission pathways, it made more sense to do an in-depth study of one disease system. It was my hope that this longitudinal study could provide valuable knowledge concerning the transmission of pathogens at the backyard chicken wild bird interface. Here, in this slide, we have multiple examples of pathogens that have either spilled over into wild birds or chickens, or they're shared between both groups, or in the case of Mycoplasma gallisepticum, have actually demonstrated a host shift and is now an endemic pathogen in members of the Fringillidae and many related passerine species. So how does pathogen transmission happen? So it requires a contact between an infectious and a susceptible host. And while contact means different things for different pathogens, for example, Newcastle disease virus is traditionally transmitted through respiratory secretions, fomites, or the fecal oral route. Thus, it includes the following pathways. For example, backyard chickens and wild birds will both exploit resources used by the other group. Wild birds will often utilize coops as shelter, and backyard chickens will utilize habitat substrates that are used by wild birds. Supplemental feed and water are used by both groups. Supplemental feed and its spillage serves as an attractant to wild birds, while bird feeders actually will attract chickens. Water is often utilized by both groups. Species such as house finches, Carolina wrens, and eastern phoebes will readily nest in structures, including chicken coops. Predation is another form of contact that can create opportunities for pathogen transmission. Chickens will readily consume eggs or other small birds, while accipiters and raptors will depredate chickens. So in reviewing chicken outbreaks, specifically Newcastle disease virus, I found that wild birds are often overlooked or under-tested in epizootics. Cross-sectional studies, which are studies which go out and examine birds at a single point in time, are insufficient to identify which wild bird species may serve as bridge hosts between multiple backyard chicken flocks. Concerning the latest Newcastle disease epizootic in California, there was a big outbreak back in 2018 and 2019. Wild birds were just not being tested for exposure to the virus. As backyard chickens become more popular and more households own them, the potential for Newcastle disease virus outbreaks increases. So as a result of my master's work and then reviewing the literature, I realized that Lasota, which is actually a live Newcastle disease virus vaccine, is an excellent model pathogen for testing transmission from backyard chickens to wild birds for several reasons. Like many avian pathogens, Lasota is a respiratory pathogen. It is presumed that all wild birds are susceptible. It's becoming increasingly known for spillover events. For example, additional papers on vaccine spillover have been published in addition to some work that we did in 2012 and through 2016. And in those studies, there has been no evidence of disease in wild birds as a result of infection with Lasota. And finally, virulent Newcastle is endemic in many countries, but it's not considered endemic in the United States. It's only considered endemic when it's being passed between chicken flock to chicken flock. However, it is found in wild birds. So we need to understand more about these dynamics. Unfortunately, we've also experienced multiple outbreaks in the last 30 years in backyard chickens, and it costs millions of dollars, and we have to depopulate millions of chickens to eradicate these outbreaks. And then the effects upon virulent Newcastle disease on our wild bird populations is unknown. So this looked like a really interesting area for research. So what do we know about Newcastle disease virus in wild birds? We know that the columbiformes, cormorants and shags, and members of the Anatidae, so our ducks, can serve as reservoirs for NDV. And NDV is short for Newcastle disease virus. NDV is not considered endemic in the United States, but it's regularly isolated from columbids and cormorants, and class 1 viruses are found in waterfowl. For the work I did in my dissertation, we didn't focus on pigeons and cormorants because those are not the species that come to chicken coops. So we weren't thinking that transmission would occur using that transmission pathway. So we investigated songbirds because their ecology concerning Newcastle disease is actually relatively unknown. Cross-sectional studies have generally focused on invasive species such as house sparrows and European starlings in the United States. But given that all wild birds are presumed susceptible, passerines have been relatively unexplored. Passerines are also highly diverse and likely have diverse susceptibilities to Newcastle disease virus. So here are the objectives I designed for the study. So for the first objective, I tested the assumption that all wild birds were susceptible to the virus in relation to Georgia's passerines. My second objective tested which species would become infected with the vaccine if they shared habitat with shedding chickens under field conditions. And my third objective tested the assumption that native birds would enter the coops, and if so, who were they? And could we measure how often they came to the coops? So as a result of my master's work and my exploratory research in conjunction with the US National Poultry Research Center, we identified these primary hypotheses as avenues of research into Newcastle disease virus pathogen transmission at the backyard chicken wild bird interface. And our first hypothesis, we selected wild bird species would be susceptible to the Lasota vaccine, both via direct and secondary exposure to infectious chickens in vivo. My second hypothesis was that wild bird species are attracted to supplemental feed, would become infected and seroconvert, which means that they would develop antibodies in response to exposure from the Lasota vaccine virus in the field. And my third hypothesis was that wild birds would be attracted to chicken coops as a result of seeing chickens eat supplemental feed. So here's a quick break. If you have backyard chickens, don't shame them because they do the best they can. So let's review my first objective, susceptibility. Identifying within family and between family susceptibilities of select taxa that have contact with backyard chickens. And when I talk about susceptibility, what do I mean? Susceptibility is the ability to become infected, right? So there's becoming infected and then there's showing evidence of disease. Those are two separate ideas. So what we were interested in was not that our wild birds would demonstrate evidence of disease because we had not seen that in the literature. We were interested in the idea that the birds would become infected such that they would be able to transmit the virus, the vaccine virus, to other birds. So given my first hypothesis, in which selected wild bird species would be susceptible to the Lasota vaccine, both through direct and secondary exposure to infectious in chickens, let's see how the data stacks. Sorry, that was my dog. The field work for the first experiment took place at 4 sites in North Georgia, in Athens and Watkinsville, over a 2-week period at chicken-free sites. Our primary objective was to capture wild birds, to transport them to the US National Poultry Research Laboratory from sites that were not associated with backyard chickens, so that they were immunologically naive. And by immunologically naive, they came from sites where they didn't already have antibodies to free-roaming Newcastle disease virus. The capture period was 2 weeks long, and each site represented a different habitat. So we represented grasslands, wetland, forested, and agricultural sites. We targeted northern cardinals, brown-headed cowbirds, American goldfinches, and house finches because they are often found in backyard chicken habitats. We also targeted chipping sparrows, but that was the only species we couldn't catch enough of. We used baited mist nets, and we captured 110 non-target individuals and 43 target individuals of our 4 species. From each wild bird, we collected blood and swabs and took standard morphometric and demographic measurements such as age, sex, and weight. Our 43 wild birds of our 4 target species for the most part represented different taxonomic families: northern cardinals in the Cardinalidae, American goldfinches of the Fringillidae, also house finches of the Fringillidae, New World finches, and brown-headed cowbirds in the Icteridae. We had a total of 13 groups, and here you can see the breakdown. The red boxes indicate the wild bird flocks in each isolator. Each species was placed into separate isolator. We had a total of 13 isolators, and there was no mixing of flocks. At the end of the 14-day acclimation period began the 14-day inoculation period. So for 14 days, they kind of hung out, and we let them acclimate to being indoors. And then at the end of that 14-day period, we either gave the wild birds the vaccine virus directly, or we inoculated the chickens that were in the isolator with them with the vaccine virus. So at 0 DPI, so at 0 days post-inoculation, certain birds were inoculated with the Lasota vaccine virus, and I'll get into more detail on that. So at 0 DPI, or 0 days post-inoculation, or the date of inoculation, we inoculated all the wild birds in the direct exposure experiment. So that's our direct inoculation experiment that you can see at the top. In our low density exposure group, we had wild birds that were exposed to 1 infectious chicken. And we had wild birds in our high density exposure experiment exposed to 2 infectious chickens. And each group was isolated using isolators so that we could measure transmission over a 14-day period. All groups were swabbed periodically during the 14-day post-inoculation period, and blood for serology was recollected at 14 days post-inoculation. So as you can see here, we had northern cardinals, brown-headed cowbirds, house finches, American goldfinches, 1 group of 5 American goldfinches, and 1 group of 2 American goldfinches, because we had a maximum number of birds that could go into each isolator. So here are the results of the direct exposure study. So we were able to detect viral shedding. So that was the purpose of this, right? Susceptibility. Were we able to recover virus from the birds that were exposed to infectious birds, or birds that were given the vaccine virus directly? So red dots indicate shedding from a specific bird on a specific date. By the end of the experiment, here you can see that viral shedding was detected sporadically in some individuals, especially amongst the American goldfinches. So this is the direct inoculation experiment. And northern cardinals, you know, they definitely shed for a long period of time, whereas you can see our house finches didn't start shedding until day 4, even day 7. And goldfinches were very intermittent. So by the end of the experiment, you can see here cardinals of our direct exposure individuals, 2 out of 2 shed virus and 2 out of 2 developed antibodies. For our brown-headed cowbirds that were directly inoculated, 3 out of 3 shed virus. So we recovered virus from 3 birds that we inoculated, but only 1 out of the 3 developed an antibody response. For our house finches, 4 out of 4 shed virus, but 0 out of 4 developed antibodies. And for our American goldfinches, 7 out of 7 shed virus, but 0 out of 7 demonstrated an antibody response. So there's definitely a differential response here to the vaccine virus, according to different species. So for our low and high density experiments, chickens had the highest viral titers and antibody titers, but they had a very short shedding period of only 4 out of 14 days. in our high density experiment, so for the birds that were exposed to 2 infectious chickens, only 1 American goldfinch out of 5 and 1 house finch out of 4 tested positive for transmission from exposure to 2 infectious chickens. None of the wild birds in the low density experiment demonstrated evidence of transmission, and all tested non-target individuals from the field were negative for antibodies. So what that says to us is they didn't come in with antibodies. And even though they were exposed to infectious chickens in the low density experiment, 1 chicken just didn't seem to generate enough virus to infect the birds in the isolators. So in the direct inoculation experiment, what we found is that while all wild bird species are susceptible to direct inoculation, so if they're directly given the vaccine, not all developed antibodies within the 14-day window. Sporadic shedding was also seen in several individuals, which has implications for cross-sectional studies that go out and identify wild birds that are actively shedding. So this is important because some birds may be in the middle of a shedding cycle, but because of intermittent shedding, when you go out and you're testing these birds for evidence of disease in the wild, they may not be shedding that day, and it may be missed. And that's really important because we're in the middle of a pandemic right now because of viral spillover. So these kinds of patterns are really important to keep in mind when going out and testing animals in the field. So another important point was that the number of shedding chickens that wild birds are exposed to may be important in Newcastle disease virus transmission dynamics. So that concluded the first experiment, and I know that was long, and I appreciate everyone's attention. So we're going to go to what is my favorite experiment that we did, which was our field experiment. Our second objective was to identify the species that are susceptible to transmission with the Newcastle disease Lasota vaccine virus when backyard chickens are shedding it. We performed this experiment at sites with different husbandry protocols to determine whether free-roaming or coop chickens had more transmission events. Specifically, we asked whether more birds would seroconvert at the free-range site than at the cooped site when they overlapped in habitat with backyard chickens. So for this experiment, I hypothesized that the wild bird species that are attracted to supplemental feed would become infected and seroconvert with the Lasota vaccine virus in the field. And I know this seems counterintuitive. I had a hard time seeing this pattern in the isolators, so why would I expect this under field conditions? And it's because we worked with more chickens under field conditions. So, but I don't want to give away the ending. I also expected some overlap between my supplemental feeders, so I call those the birds that come in and eat the feed in the chicken coops, and my Cornell feeder watch species, and I'll explain what that is. So for those of you that don't know, Cornell Feeder Watch is a winter-long survey of birds that visit feeders at backyards, nature centers, community areas, and other locations in North America. Participants periodically count the birds they see at their feeders and send their counts to Project Feeder Watch. On this page, you can see the species that citizen scientists have identified as feeder watch birds in the Southeast. And we'll revisit this slide once we've reviewed the results for this experiment. So this is a broad view of our sites in North Georgia, spanning Jackson, Oconee, and Clark County, which are juxtaposed upon one another. They're well, they're not interconnected, but they're connected to one another. There were 3 sites. Site S, the chickens were cooped, and there were 9 of them. At site L, the chickens free roamed, and there were 15 of them. And site C had no chickens, but there was a coop. So because we wanted to ensure that our avian communities were independent of one another, they were at least 10 kilometers apart. And if you focus and you look closely here, you can see a mist net set up right in front of the chicken coop. And you can see another mist net over here with the pink bungee cord. So at Site C, where there were no chickens, we took blood and swab from wild birds for 30 days. And that was our capture period. But at our sites where we had chickens, we divided our sampling period into a pre-inoculation phase, an inoculation phase, which was the date that we gave the vaccine virus to the chickens, and then a post-inoculation phase where we caught the wild birds in the area for 3 weeks beforehand. And we did this pre-inoculation phase because we wanted to measure how much Newcastle disease virus exposure in terms of antibodies the birds had prior to giving the vaccine virus to the chickens and then measuring transmission. And then our post-inoculation phase was clearly to measure chicken transmission. So on the date of inoculation, each chicken was given the same dose. And in the wild birds, during the pre-inoculation and the post-inoculation phase, we collected oral swabs and blood for serology whenever possible. And serology is for antibody testing. We also collected standard morphometric and demographic data, such as age, sex, and body mass. We used what are called hemagglutination inhibition tests, so the type of antibody test, to evaluate chicken and wild bird serology, and oral swabs were evaluated using real-time PCR. And as you can see at the bottom, these are my wonderful undergraduates who are helping build one of the coops. And there's an undergrad holding James, holding a rooster. And there we are swabbing a gray catbird. And I'm there holding a ruby crown kinglet. So here are the results. At Site C, where we had no chickens, we caught 128 wild birds in 30 days, which was amazing. And we primarily caught tufted titmice, northern cardinals, and Carolina wrens. And you can see northern cardinals are going to play a big role here. During the pre-inoculation phase at Site S, where our chickens were cooped, we caught 45 wild birds, and we caught primarily tufted titmice, northern cardinals, and Carolina wrens. And at Site L, where our chickens free-ranged, we caught primarily tufted titmice, Carolina wrens, and a lot of downy woodpeckers, which I found really interesting. During the post-inoculation phase, at Site S, where our chickens were cooped, we caught Carolina wrens, tufted titmice, and ruby crown kinglets. We caught, as you can see, a lot more birds at Site S than we caught at Site L, where we caught 56 wild birds at the post-inoculation phase. So after the chickens had been given the vaccine virus, we caught primarily pine warblers, myrtle warblers, and eastern bluebirds. So very quickly, here are some results. At Site C, where we had no chickens, but we had a coop, 0% of our birds developed antibodies or had antibodies. And that was as we expected. During the pre-inoculation phase, interestingly enough, at Site S, where our chickens were cooped, 36.8% had antibodies. And at Site L, where our chickens free-ranged, 66.6% had antibodies for Newcastle disease. This is before we released the vaccine into the chickens. So at Site S, after we gave the vaccine to the chickens, that number doubled, and 70% were positive for antibodies, which was evidence that transmission had occurred. And at Site L, interestingly enough, with our free-range chickens, there was no change in the number of birds with antibodies. So that's interesting. This slide, very quickly, as you can see here, These are our chickens. So we also tested antibodies in our chickens to make sure that they seroconverted or they developed antibodies to the vaccine. And as you can see in the pre-inoculation phase, our free-range chickens are in red, and our coop chickens are in blue. And our coop chickens developed a much higher antibody response over the course of 18 days than our cooped chickens, or I'm sorry, our free-range chickens. So even though our free-range chickens, free-ranged and had engaged in more activity, we actually saw a greater antibody response in our coop chickens, and I found that to be really interesting. So for wild birds, as you can see in this graph, it appears that our titers, which is the level of antibodies that are measurable, more birds, or titers were higher for birds at site L, which is the opposite of the chickens. So the birds had higher antibody levels at site L, which is where our chickens free ranged. But at site S, more birds ended up seroconverting. We ran a linear mixed model and found that there was no statistically significant difference in titers between site L and site S. But there was a statistically significant difference when compared to site C, which makes sense because all of our birds at site C, where we had a coop, but we had no chickens, none of the birds had antibodies to Newcastle disease virus. The most important thing to take away from this is that we identified a total of 13 species that seroconverted or developed antibodies in response to exposure to shedding chickens. Because of sampling sizes, only 10 are shown in this graph. Of the real-time PCR that was completed, we have some exciting preliminary results. 4 of 28 birds that were tested, or 14.3%, were positive for Newcastle disease virus RNA, according to real-time PCR. I'm really excited to have all these PCR results, but due to COVID restrictions, the labs all shut down the semester before I defended my dissertation. So we're still trying to figure out the logistics of me finishing my PCR. And so if you remember our feeder watch species, I've highlighted those in pink, those that were overlapped with our study. We're missing one species, eastern phoebes. What's interesting is that a lot of these species are primarily ground foragers or mid-story foragers, and they're either seed eaters or omnivores. Several species that here in this in this image were captured at high frequencies, such as pine warblers, but none of them seroconverted. So that's also an interesting result. In short, chipping sparrows, northern cardinals, tufted titmice had the highest antibody responses across all 3 sites. And so if you look at antibody responses being related to susceptibility, that means that they could have had the highest exposures. I mean, it's possible, but we needed to do an additional experiment to really look at that. We'll talk about that in a moment. So why did we not see a lot of transmission in our isolator birds, but we saw transmission in the field? There were isolator and IACUC limitations that may have made transmission difficult during the laboratory experiment, including the number of shedding chickens. And I keep going back to that, how density is important. Our results do provide evidence that Newcastle disease virus spillover between passerines, specifically Cornell feeder watch species, and backyard chickens does occur. While I was primarily supported in the idea that birds who seroconverted were Cornell feeder watch species, there were many listed as feeder watch birds that did not seroconvert. So that appears to be an avenue for future research. And just so that everyone knows, I did have to swab and catch those chickens and take blood from those chickens, and catching a chicken is really hard. So yeah, that was probably the hardest part of my dissertation. Our final objective was to identify and quantify the species that enter the backyard chicken coops and how often they do so. So we caught birds in mist nets for our second experiment, and we identified which ones, increased in antibodies and which ones did not increase in antibodies over time as a result of, being exposed to these infectious chickens in their habitat. But now we wanted to look at who, in terms of species of wild birds, was going into the coops and how often they were doing so. So let's see how this last hypothesis, which is, I hypothesized that wild birds would be attracted to the chicken coops as a result of seeing chickens eat supplemental feed. Let's see how that stacked up. So as you can see, we performed this experiment at the same sites where we performed our field transmission experiment. The same rules applied. We had a site with cooped chickens, free-ranging chickens, and no chickens. So we performed hour-long observations, and these comprised of systematic observations at these 3 sites associated with backyard chickens between February and May of 2018. So Site C served as the control, which was a coop without chickens. Site S cooped their chickens during the day, and Site L, they free roamed during the day. And all of these coops, regardless of whether or not there were chickens present, were provided with supplemental feed. Food was added to the pens, which consisted of a wire mesh surrounding each coop instead of to the coop interiors, so that during observations, we could identify the species that entered the pens and the frequency with which they did so. Each site was observed 2 times per week at opportunistic times for an hour at each observation point. Different distance classes were recorded, which I will get into with further detail. This schematic illustrates how observations were performed at each farm. As you can see, the chicken coop is in the middle, and each DX corresponds to a distance class. Birds were identified by sight and by sound. D1 represents the inside of the coop in the pen. D2 is the edge of the pen. I'm so sorry, that's my dog. The edge of the pen to 10 meters. D3 is 10 to 25 meters from the coop perimeter. D4 is 25 to 50 meters. And D5 is greater than 50 meters from the coop. So the black arrow denotes the observer point of view. We observe the observations as a modified point count to not only identify who was going into the coop, but also to identify what species were in the same habitat and were actually not going into the coops. We use the following equations, and I'm not going to go into them, so don't worry, to calculate the contact rate and the visit rate for each species in D1 and D2. The contact rate was defined as the mean number of visits per observation within a 60-minute period. The visit rate was calculated as the mean number of visits over a 12-hour period, or 720 minutes, since these are diurnal passerines. So over 60-hour-long observations performed between February and May of 2018, our mean detection rate was 26 wild birds per observation period. The species richness was 12.7, and the mean number of families was 9.3. Based on visits to D1, which, as you recall, was the distance class that correlated to the inside of the coop, we identified 14 species that were high risk for pathogen exposure. These 14 species had a total of 589 contacts, which we defined a contact as entering just D1 or D1 and D2. And lastly, these 14 species were the most frequently detected overall, comprising 67% of all detections across all distance classes. So over 60 hours of observation, 1,574 individual wild birds were detected across 3 sites in northern Georgia, comprising 72 species over the field season from 24 families. Across all sites, tufted titmice, again, we get with the tufted titmice, northern cardinals, Carolina chickadees, and Carolina wrens were the 4 most commonly detected species. Chipping sparrows, as you can see in the graph, had a high detection rate, but only at one site, which was Site S. Site S, which, as you recall, is our cooped flock, had the greatest number of overall detections, as well as the greatest species richness. Site L, the free-ranging flock, had the fewest number of overall detections and the lowest species richness. And Site C, which was our coop with food, with supplemental food, but without chickens, was in the middle. So that's a really interesting result. The highest contact rates, so remember we defined a contact as entering D1 or D1 and D2, detected at Farm S, followed by Farm C, and then Farm L. As you can see, chipping sparrows had the highest contact rates of any species at a site, followed by tufted titmice at Farm C. So down here, tufted titmice. And myrtle warblers had the highest contact rates at Farm L. So using our contact rates, we were then able to extrapolate the mean number of visits per day. So what does all this math mean? So in essence, it means that at Site S, chipping sparrows made an average of 85.3 visits per day. Tufted titmice averaged 41.4 visits to Site S, where the chickens were cooped, and 45.3 visits to Site C, where we had no chickens. Carolina chickadees also had a high visit rate. So this is the same graph, it's just kind of a little bigger. Carolina chickadees had a high visit rate to Site C, where we had no chickens. And at Site L, where the chickens free roamed, the most frequent flyer was the myrtle warbler with an average of 15 visits per day. So let's see how my hypothesis held up. Clearly, Farm S had the highest number of visits when all birds were combined. However, given the number of visits to Farm C, where there were no chickens, but there was supplemental feed, yes, the birds were attracted to supplemental feed, but it wasn't the chickens that cued them in. So my hypothesis was not really supported. Instead, our results suggest that the role of husbandry in terms of whether or not the chickens free roamed or the chickens are cooped was not really a factor in the number of daily bird visits. It was actually secondary to the role of site species richness, which we calculated as the number of birds that visited the coops and those that did not visit the coops. It was the total birds in the entire habitat. In addition, when it came to wild bird visits, in essence, the birds weren't interested in the chickens at all. They didn't really interact with them. It was all about the food. So in summary, Farm S had the highest number of visits when all birds were combined, but given, sorry, I lost my place. So, yeah. And so this is the last slide. I hope you enjoyed all that, the math and the misnets and everything that was involved with my dissertation. And here are some limitations of, in case anyone has any questions, here are some limitations of what we examined. So seasonality. In experiment 1, experiment 2, and experiment 3, they were performed during different seasons. So it was difficult to compare avian communities across the 3 experiments. So we definitely had 3 separate experiments and tried not to pool data between all of them. For our laboratory experiment, what we found was that it's quite possible that holding birds for 14 days post-inoculation may have just been too soon, that we may have needed to hold on to them for a longer period of time to see if they would have developed antibodies, especially our house finches and American goldfinches. We had free-roaming cats at site L. There was a low recapture rate and a low visit rate, so that may have impacted some of the species richness that we saw. Increases in antibody titers versus those that seroconverted. So we did see birds that increased in antibody titers over time, and that gave us an idea, that gave us the knowledge that that transmission did occur, and that antibodies increased as a result of viral exposure from infectious chickens. Birds that were positive for antibodies at the field sites with the chickens before we actually gave the vaccine to the chickens, that complicated the analysis somewhat, and that's an avenue for future research as to why they were positive for antibodies there before we disseminated the vaccine. And we didn't have a coop without food in experiment 3, and that may have actually given us a better idea of abundance and richness at the other sites for comparison. So a few novel results: intermittent shedding from house finches and goldfinches. There are likely poor sentinels for Newcastle disease virus, especially where Newcastle disease virus happens often in the western part of the country. We identified 14 high-risk frequent flyers in experiment 3 and 13 species that seroconverted in experiment 2. So there was a high degree of overlap there. And that's really interesting. Really, my dissertation, 6.5 years, came down to food, food, food. That was really the big takeaway from this. Tufted titmice, chipping sparrows, and northern cardinals developed the highest antibody titers, and that suggests that they may make excellent surveillance cross-sectional study targets for this particular pathogen. And in general, northern cardinals are excellent sentinels for NDB across all experiments. Here's some take-home points. Passerines just differ in their susceptibility to Newcastle disease virus, and more research is needed. The chickens all shed the vaccine virus, which we expected them to, and 13 of our songbirds seroconverted in experiment 2. Our PCR is pending, and we definitely need more research into this system. And it's crucial to identify the species that come to backyard chicken coops and measure their susceptibility to NDB, especially in different geographical areas, because they may be visiting multiple backyard chicken flocks. So friends don't let friends put out communal feeders. And with that, I'd like to thank my funders who generously made this research possible: the Ford Foundation, the USDA, the Georgia Ornithological Society, the Coney Rivers Audubon Society, the Association of University Women, the College of Vet Med, the UGA Grad School, the Poultry Respiratory Disease Coordinated Agricultural Project. And I'd like to thank these amazing people who all took part in this experiment in some way, shape, or form and made it possible. And with that, I'm happy to take questions.
Great. presentation, Andrea. Very interesting.
Thank you. I'm so sorry that Patches kept falling over.
Oh, that's okay. He wanted to be the star of the show. I do have a couple questions for you, and then Larry, I'll ask some, and then you can ask some. One of the questions for people that have backyard chickens, what are some of the signs or indications that they may have developed Newcastle disease, and what can they do if they notice their chickens have some of the symptoms?
So with the vaccine, as I mentioned, there's in general not going to be any signs of disease, thankfully, and that's in wild birds. For chickens, you may get generalized respiratory signs. So it'll look like your bird has a mild case of the flu. Virulent Newcastle disease, which is kind of like high path avian influenza in the sense of that's how much it attacks the entire system of the bird. You generally, they don't make it, basically. So you have extreme lethargy, ruffled feathers, and they end up not making it. So it's a terrible pathogen, especially the virulent form, and we'd like to keep it out of our birds as much as possible.
Yes, definitely. And do you recommend people that are considering or even have backyard chickens, they do inoculate them for safety?
Yes, absolutely.
Great.
Absolutely.
Yeah. That's great advice. Do you have any questions, Larry?
In the let's see, the birds, one of your populations, you found wild birds that already had a high percentage of Newcastle disease infection, didn't you?
Yeah. So in our free roaming flock, they actually demonstrated antibodies to Newcastle disease virus. And then after I gave the vaccine to the chickens, there was no change in the percentage of birds that had antibodies to Newcastle disease virus. So how these birds had been exposed to the virus beforehand, I don't really know.
Refresh my memory, was that the wild birds or the chickens that had the Newcastle?
Yes. So the chickens all had pre-existing antibodies because they had been inoculated at birth.
Oh, okay.
Once they hatched.
Okay, explains that.
So,
okay.
Yeah. But then the wild birds also had antibodies to Newcastle disease, and we're not sure how that happened. One hypothesis, and I'm just going to postulate this here, is that so for a lot of hatcheries, when you order chickens, you get them at 1 day of age, right? And so they're vaccinated at 1 day of age. And if you remember from my experiment 1, chickens will shed Newcastle disease virus vaccine for up to 4 days. So that period that they're going through the post office, they're actually potentially still shedding virus all the way up to when they get to your house. So that might be a way that the vaccine virus is getting into the environment.
And there's the possibility perhaps that there are other chicken coop populations, not in your experiment, but the birds, if they were migrating or coming from other regions, they could have picked it up elsewhere, most likely.
Oh, yes. Like I said, 78 countries reported Newcastle disease outbreaks in 2018. I'm not sure what the number was in 2020. But yeah, migration we definitely saw antibodies in, I think I had an Eastern peewee that had a really high antibody titer. And this was, during a period of migration that Yeah.
Yes. And I did have another question. You mentioned that and I was taking notes, so bear with me,
No, you're
that
fine.
you found that the cooped, for the second part of your experiment, the cooped had a higher antibody response, and more birds were seroconverted. The cooped ones were more seroconverted than the free-ranging one. Do you have any ideas as why that may be?
I have absolutely no idea. What I think could possibly have been a factor is that there was just a higher species richness. There were just more birds at the site where the chickens were cooped. And so I think there was a greater opportunity for more birds to come in. We caught more birds. More birds came in, more birds ate, more birds interacted with the shedding chickens. So I think that's quite possibly what might have happened and what we picked up on. So biodiversity and infectious chickens don't mix.
Yes. And you most likely said this, but I cannot recall. When did you have your field seasons? What year?
Oh, so 2016, we did 2017, we did the lab and the hybrid lab field experiment. And then we did and that was in the spring. In the fall, through the winter, we did the experimental coop. We did the field inoculation experiment. And then in 2018, the following winter going into spring, we did the observation experiment.
Okay, excellent. Very interesting.
I have a question. What is the main problem that you see though? Is it that chickens will get infected by wild birds, or that wild birds will get infected by chickens?
Well, I mean, it's bidirectional, right? So any one of those individuals can likely infect another individual if contact happens. Mm-hmm. But the biggest issue I see is for kind of like the way that the analogy I like to make is, so kind of like how we had the viral spillover of the coronavirus into the human population, I'm worried about a host shift of a pathogen that is evolutionarily, I want to say, tied to chickens, that will spill over into wild birds and then become adapted to wild birds.
Right. Okay.
So that happened with mycoplasma. Right. With commercial chickens. And so I'm worried about another pathogen happening, that as people get more backyard chickens, we're going to have more contacts. We're going to have more opportunities for that spillover to happen.
Right. That one mutation that proves disastrous. Yep.
Yeah. Yeah. And it can happen it can be a spillover into a species that is not really, adapted for it, and then it moves into a species that,
right,
can be adapted to it, and then you have an issue. And with these feeder birds, they're hopping from flock to flock, so I call them promiscuous. So they're hopping from flock to flock. So not only are they passing it from flock to flock, they can pass it very easily from bird to bird. So birds that are visiting multiple flocks have multiple routes of becoming infected.
And is there any work being done to possibly inoculate wild birds against Newcastle?
So that was something that was discussed, but then I graduated. So at this moment, I am not aware of it, but of any work that's being done to inoculate wild birds. This was actually the first experiment that looked at field transmission of this vaccine virus. So I would be really interested if out west, where they had these outbreaks of Newcastle disease, that's virulent, if they would look at that and measure it and see if it was actually beneficial to these species.
Yes, great point. And now I know your study focused on backyard chickens, but I have to add, is this a serious issue with the bigger chicken farms, factory farm type chickens with huge populations? Are they having Newcastle disease issues on the East Coast?
So on the East Coast, I actually do not think we've ever had an outbreak of virulent Newcastle disease. I think there have been incidences of low virulent Newcastle disease, not in factory farms, but in wild birds. This seems to be more of an issue out west, and I'm actually not quite sure why.
Okay, excellent. And I did want to add, someone did comment in the YouTube, excellent. So you presented wonderful information. And the good thing about having our programs on YouTube recorded, we can watch it again because it was so much good information. I think this is important research that you participated in. And there's still more questions to answer. So I'm interested in your PCR results when you get them.
I am too. I am very interested in the PCR results. And I'm actually disappointed about the pandemic, like shutting down, access to the lab and access to the PCR. So that was, it's one thing to extrapolate information from antibody titers. It's another thing to recover the actual pathogen. So that's kind of like the gold star right there. So yeah.
Yes. And I did notice, if anybody wants to get in touch with you and they have further questions, how can they reach out to you?
So I can be reached at andrea.ayala@yale.edu. That's my email. I can also be reached at ayala1@uga.edu. So both of those emails are active. And I'm happy to speak with folks further. This is something I'm very passionate about. And I look forward to speaking with the audience some more, if that's possible.
Hey, Andrea,
Excellent.
Here's an old friend of yours.
How did you maintain the logo so like brightly colored? Mine are all faded.
I haven't worn this T-shirt too much over the years, but I rediscovered it about a year ago, and I've been wearing it a fair amount.
Oh, that's wonderful.
So this is our mascot. Andrea was a co-founder of the environmental club at Valencia College back around the year 2001, and was president for the first couple of years. And this was our mascot. It was a gopher tortoise, because we did have
experience of my life. And it laid the foundation for everything in my scientific career. So thank you for your mentorship, Larry. It was just, I don't have words. I mean, as you can tell, even Patches was beside herself.
It's been a long haul. It's been about 20 years. And so Andrea she was an environmental crusader. We got a lot of changes done on West Campus of Valencia College. And so you have a legacy there.
Thank you. Well, and thank you for being this amazing mentor. It wouldn't have happened without you. So thank you. So thank you.
I love this. I'm so happy that Larry suggested having you, Andrea. Your work is amazing. And I didn't know about your involvement at the Valencia campus. So I'm a Valencia alumna. So that is awesome to hear that as well.
Yeah, that's great. Thank you so much. Thank you for having me. And I'm working on, so hopefully, with my Yale work, working on cholera in waterfowl in Florida. So hopefully I'll get the chance to come back and talk to you guys again. That would be awesome.
Okay.
Absolutely. And what I will do, everyone, is put Andrea's email addresses in the description. So if you wanted to reach out to her, it's easy to find. But thank you very much for coming on today, Larry. You're always appreciated. And Andrea, thank you so much for joining us. Wonderful talk. And everyone, be safe and stay tuned for our program next month. Take care, everyone.
Bye-bye. See you, Andrea.
Bye, Larry.